5. Microbial physiology. The growth may be defined as an increase in cellular constituent. It leads to rise in cell number when microorganisms reproduce by process like budding or binary fission. In the latter, individual cells enlarge and divide to yield two progenies of approximately equal size. The growth also results when the cell simply becomes longer or larger. If the microorganism is enocytic, that is, multi-nucleated organisms in which the nuclear divisions are not accompanied by cell divisions. The growth results in an increase in cell size but not cell number. It is usually not convenient to investigate the growth and reproductions of individual microorganisms because of their small size. Therefore, when studying growth, the microbiologists normally follow the changes in total population number. 5.1. The growth curve. The population growth is studied by analyzing the growth curve of microbial culture. When microorganisms are cultivated in a liquid medium, they usually are grown in a batch culture or closed system. That is, they are incubated in a closed culture vessel with a single batch of medium. Because no fresh medium is provided during the incubation, the nutrient concentration declines and the concentration of waste increases. The growth of microorganism reproducing by the binary fission can be plotted as a logarithmic of a number of viable cells versus the incubation time. The resulting curve has four distinct phases: lag phase, log phase, stationary phase, and death phase. Lag phase. When microorganisms are introduced into the culture medium, usually no immediate increase in cell number occurs, and therefore this period is called as a lag phase. Exponential phase. During the exponential or log phase, the microorganisms are growing and dividing at a maximal rate possible, giving their genetic potential, the number of the medium, and the condition under which they are growing. Their rate of growth is constant during the exponential phase. That is, the microorganisms are dividing and doubling in number at regular intervals. Stationary phase. Eventually, the population growth ceases and the growth curve becomes horizontal. This stationary phase usually is attained by bacteria at a population level of around 109 cells per ml. Other microorganisms normally do not reach such high population density. The protozoan and algae culture often have maximum concentration of about 106 cell per ml. Death phase. The detrimental environmental changes like nutrient deprivations and the buildup of toxic waste leads to the decline in a number of viable cells. Death phase. The detrimental environmental changes like nutrient deprivations and the buildup of toxic waste leads to the decline in the number of viable cell. Characteristics of death phase. The death of the microbial populations, like its growth during the exponential phase, is usually logarithmic. That is, a constant proportions of cell dies every hour. Strategies of cell divisions. The flow of metabolism and timing of individuals' activities by intricately coordinated so that it leads to the orderly process of cell duplications, the process that takes place during the cell cycle, or the coordinations of chromosome replication with the formation of a division septum. The production and activity of various protein essential to the cell division processes and the regulations of these events. The cell wall peptidoglycan of E. coli and other gram-negative bacteria is extended by a process of intercalation of a new wall material by a number of enzymes. At a point, a series of events results in the assembly of large group of proteins into a ring, where a constriction occurs between two daughter cells. This ring structure and its component parts are called a division. The cell division can be inhibited without disrupting the continued linear elongation of the cells. The action of certain lactam antibiotics directly affects the process of cell division, can result in the formation of filaments lacking septation. The proposed step in the cell division in E. coli involves three main groups of enzymes. 1. The first group contains enzymes that forms the peptidoglycan saculas around the growing cells. In the absence of modifying activities, this set of enzymes produces the minimum amount of peptidoglycan needed to surround the cell. That is, it produces a spherical cell that enlarges without divisions too. The second group of modifying reactions contains, for example, the rod A PBP2 system that results in the development of cylindrical shape of a peptidoglycan saculus. These functions must be active over the entire surface except at the poles, because the pre-existing cylindrical surfaces are converted to the spherical surface after inactivations of either of these two gene products. Also, a spherical surface formed in the absence of these product is replaced by or converted to a cylindrical surface after reactivation of this system. 3. The third group, responsible for cell division through the formation of new septation site, acts periodically, and it confined to the site of septations at the cell surface. A switch to formation of type of peptidoglycan murin required for septation occurs late in the E. coli cell cycle. Inhibition of a beta-lactam agent gives rise to long multinucleated non-separate filament. The switching from cell elongation to septum formation may result from the change in the relative availability of different peptide chains required by the two competing systems. The figure, the role of cell division component in E. coli conceptual. The process begins with the assembly of FT Z at the midpoint of the cell and target min E to the same locations. This penicillin binding protein FTS1 is a late recurrent to the division site, and its septal location is dependent on interactions with the FTZZ, FTS A, FTSQ, FTSL, as well as intract membrane anchor. The FTSZ protein acts at an early stage of the septation process. The FTSZ is a magnesium-dependent GTPase dispersed throughout the cell. Just prior to the onset of special invagination, the FTSZ is required from the cytoplasm to the division site. The cell division does not take place until a round of replication has been completed. Cell division is triggered by events that occurs at or near the termination of replication. The most important of these is deconcatenations of replicated chromosomes. The separated chromosomes are then participated into the two daughter cells. The division occurs subsequent to or simultaneously with partitioning. In E. coli, several enzymes appears to participate in resolving chromosomes dimer formed during the replication. The two site-specific recombinase XCRC and XERD act in concert at a site near the terminus of chromosomal replication DIF to separate chromosomes dimer into monomer during the process of cell division. Stress response. The ability of microbes to stress and respond correctly to impromptu alteration in the environment is crucial to their survival. The condition that allows the maximum growth rate outside the laboratory are few and far between. As a result, most bacteria live in a concentrator of stress. The osmotic stress and osmoregulation. Many bacteria hypertonic or hyperosmotic conditions results in water loss from the cytoplasm, causes the cell to shrink. Plasmolysis. The hypotonic or hypoosmotic condition results in an influx of water into the cytoplasm, which causes the cell to swell. Plasmoptosis and perhaps burst in a process referred to as osmotic lysis. The microbial membrane are freely permeable to water, and therefore the water inside the cell is essential at equilibrium with the outside of the cell. The microbes with a cell wall can and do maintain a high cytoplasmic solute concentration relative to the outside. The most rapid response to this osmotic up shock is an increase in K+ ion influx through the at least three uptake system in E. coli, Salmonella, Enterica, and other bacteria. Many microbes also accumulate the disaccharide trihalose composed of two glucose residue as a compatible solute in response to the osmotic stress as well as the starvation, the thermal stress, and desiccation stress. The complex sugar known as a membrane-derived oligosaccharide found in the peritplasm of gram-negative bacteria can reduce by growth in conditions of low osmolality. Aerob to anaerobic transition. The aerob to anaerobic transition, the facultative microorganisms such as E. coli and Streptococcus enterica can modify their metabolism to accommodate growth under either aerobic or anaerobic condition. The transition between the aerobic and anaerobic metabolism is accompanied by alteration in the rate, route, and efficiency of pathway of electron flow. Under anaerobic conditions, without alternate electron acceptors, the pyruvate is converted to formate acetate or ethanol, carbon dioxide, and hydrogen gas. Obligate anaerobe lack both superoxide dismutase and catalase that deals with reactive oxygen species. Thus, oxygen is toxic to them. Example: Clostridium microaerophils capnophiles only grow in low concentration of oxygen, 21%, like Campylobacter jejuni, Neisseria gonorrhea, and Streptococcus. Most bacteria grow in range between 0.85% sodium chloride physiological saline and 3.5% sodium chloride seawater. Most microorganisms are halophile, like E. coli, in salt intolerant. The halophile grow in extreme high salt environment, 15 to 30% salt. The halobacterium grow best in the environment having 18% salt concentration. The acidophile lactobacillus thiooxidants maintain an internal pH of 6.5 over an external range of 1 to 5.0. The neutrophils E. coli cannot withstand pH condition greater than 8 or below 4.5 alkalophil. The bacillus alkaligens fecalis and Vibrio cholera maintains internal pH of 9.5 over on extreme pH range 9 to 11. The thermal stress and the heat shock response upon a shift from 30 to 42 WC, the E. coli and other bacteria transitionally increases the rate of synthesis of a set of protein called heat shock protein. Many of these heat shock proteins are required for cell growth or survival at more elevated temperature thermotolerance. There are nearly 50 heat shock inducible protein identified in E. coli. They can subdivide into those regulated by alternative factors and two component regulatory systems. The Bacillus subtilis has 4 classes of heat shock genes. All of them are not described here, but one is particularly interesting because of mechanism used to control the genes. The class 1 heat shock genes includes the major chaperone, DNA-K, DNA-J, GRPE, and GROES. The psychrophilic yeast Leucosporidium grows well at -1 kc. In these organisms, there is a positive correlation between the growth temperature and unsaturated fatty acid composition of the cell lipids. The psychrophilic bacterium Micrococcus cryophilus also undergo alteration in its lipid composition with changes in growth temperature.